Hello, everyone. Well, thank you, John. Thank you, Carrie, for the invitation. Hello, everyone in the audience, too. My name is Leonardo Mena-Rivera. I'm a postdoctoral researcher at the University of Oxford, working with Professor Robert Hilton. And today, I'm presenting some preliminary data from our Riverscape project that is funded by the European Research Council with additional support of PP Systems. Just in general, the aims of the project are to better constrain the drivers of greenhouse gas emissions from rivers. Sorry. In particular, well, my work is more focused on assessing how susceptible organic carbon is to degradation once it moves from the terrestrial to the fluvial system. So, well, the Arctic region is important in regulating global climate and global biochemical cycles, with Arctic soils accumulating almost twice of the carbon present in the atmosphere. So the current estimate indicates that there is 177 petagrams of carbon in permafrost soils and non-permafrost soils. And they are distributed not equally across the Arctic. Let me see if I can just show the spotlight. Can you see it? Yes. They are not distributed equally across the Arctic, and there are regions with continuous permafrost, discontinuous, or sporadic permafrost. As I guess you know, the Arctic has been warming nearly 4 times faster than the mean global rate. And this figure at the top right shows an increase in temperature since 2000, since the year 2000, that is also related to a decrease in sea ice cover almost at the same time. And these warming-induced changes can impact the physical and biological degradation of organic matter across the landscape, the hydrological connectivity, and also they can impact sediment transport and sediment dynamics in general. Now, the magnitude of these impacts are quite complex to assess and predict, given some local and regional conditions and processes. So as temperature continues to increase and permafrost thaw, some of these carbon stocks can become more exposed to degradation and mobilization. And potentially they can release greenhouse gases such as methane or carbon dioxide, therefore acting as a positive feedback for future warming. But they can also impact the cycles of other elements such as nitrogen, sulfur, and mercury, which are tightly coupled to the carbon cycle. In order to quantify the long-term impacts of this mobilization of carbon and degradation of carbon, we need to assess how susceptible these carbon stocks are to degradation. And this remains challenging, and two approaches have been using in the past. The first one is, well, some scientists have been using long-term incubations. Well, they go to the field, collect permafrost soils, and incubate it in the lab for years. We're talking here about decadal timescales, and measure greenhouse gas releases. So in this figure at the top, you can see the increase in carbon release as a gas over time as a function of time at higher temperatures. The other approach is to use warming experiments in the field. So these are underground heating experiments, and then also over months, over several months and weeks, and to measure how warming increases greenhouse gas releases. as temperature increases. So a different approach is to use river sediments to trace changes in organic carbon inputs across the landscapes. That's what we're trying to do. That's what I'm trying to do in this project. In particular, we want to use thermal analysis to assess the stability and reactivity of the organic carbon presence in sediments. We use river sediments because they contain a integrated signal of different sources of organic carbon and degradation states. So we're using a technique that is called ramp temperature oxidation or pyrolysis. There are, it's also can be called different names, thermal ramp oxidation or pyroxidation, but it basically consists in just heating a sediment or a solid sample over a constant temperature gradient, for example, from 100 to 1000 degrees at a rate of 5 degrees per minute. And as the sample is heated and decomposed, we can measure the CO2 that is produced over time. And during this process or during that degradation is related to the chemical composition of the sample, the bond structure, and of course, the environment. So in terms of instrumentation, there are limited options available in the market at the moment to run this type of analysis. An instrument includes three main steps: combustion, oxidation, CO2 measurement over time, and CO2 collection for stable and radiocarbon analysis. And this is an example in the United States. The first part of the instrument is a reactor that where we can use a ramp temperature. Then there's a second reactor that works at a constant temperature, and then an infrared CO2 analyzer where we can measure a CO2 concentration over time. Then at the end, we have a couple of cryogenic traps to collect that gas, purify it, and use it for stable carbon and radiocarbon analysis. So this technique really provides information about chemical structure and age. I think it works very well because it can give you two separate sources of information, and it closes the gaps between the gap between bulk and molecular level analysis. And one of the limitations of bulk analysis is that well, it's a bulk sample, so a lot of information is lost during the analysis. And in the other side, molecular level analysis are sometimes time consuming and they require quite a lot of sample amount. So with ramp temperature oxidation, they can give you, it's a good, it's just in the middle, and then give you some advantages in terms of information and sample processing. So part of my work here at Oxford was being to develop an instrument to run this type of analysis. So our instrument works in oxidation mode, uses cryogenic traps, and we measure CO2 concentrations using a PP Systems SBA5 analyzer. So just in general, there's a gas supply, which at the moment for just thermal analysis, we can use zero-grade air. For radiocarbon, we could use synthetic air instead of helium. And then we have two carbonite gyro-furnaces, one that works in a ramp from 100 to 950 degrees at 5 degrees per minute, 10 degrees per minute, or 15 degrees per minute. Then we have a second oxidation, a second oven where there are some reagents just for oxidation and purification of the gases, water traps, naphthalene and magnesium perchloride, and the infrared analyzer that we're using the PP systems in a range of 0 to 10,000 ppb V. And then we're also developing this part of the instrument at the moment, which is a semi-automatic system to collect and purify CO2 for stable and radiocarbon analysis. So just to show you a picture, this is how the instrument looks like. Everything is, well, most is custom made, right, and custom built here in the university. We have the cylinder with the gas, a filter to purify the air, vacuum connections, the SBA5 analyzer, and we also have a custom-built software to measure the output from the SBA5 analyzer. Sorry, the output of the SBA5 analyzer and integrate it with the temperature measurements that we recorded using thermocouples and an Arduino microcontroller. So in terms of the instrument works pretty well. In terms of ramp temperature, we can measure ramp temperatures with a resolution of 0.1 or with a deviation of 0.1 degrees per minute. And during thermograms of soil standards or reference materials, we see differences in a maximum temperature of plus minus 5 degrees. So this is the output that the instrument gives you, CO2 concentration over temperature or over time. And then we can use that information, this information of the thermograms, to calculate some probability distributions of activation energy. So this is a metric I'm going to explain a little bit later, but it's basically related to organic matter stability, organic carbon stability, and chemical diversity. Although this instrument is not designed to measure organic carbon concentration like as equivalent to TOC, it works pretty well. We can measure carbon content in our samples with an error of 1%. However, we don't use it with that to do that, right? And we use a TOC analyzer for that, but still the measurements that we are getting from the CO2 analyzer are reliable enough to measure organic carbon in samples, basically. So once we develop and build the instrument, we've been analyzing sediments from the Mackenzie River in Canada. We focus on these catchments for several reasons. One of the main ones is because it's the largest river catchment in Canada with an area of 1.8 million square, which is nearly kilometers square, which is nearly 20% of the land of Canada. Also, it's the fourth largest source of fresh water to the Arctic Ocean and the man-made sediment supplier, and it has a flux of nearly 2.2 million tons per year of particular organic carbon, which is among the highest in the Arctic. Now, this region in particular has presented an increase in temperature of nearly 3%, 3 degrees, sorry, since the mid-20th century. And that's been related with an increase in suspended sediment discharge, 46% from 2005 to 2015, and also an increase in the flux of dissolved organic carbon of 39% from 1972 to 2012. So in terms of land use and permafrost cover, most of the catchment is covered by continuous and this continuous permafrost, as you can see here. And we've been working in five main sites: the Delta region, the Mackenzie River, which is the main river, and the Arctic and Peel tributaries, and also the Liard, which is one of the main rivers that delivers sediments to the Mackenzie. So in terms of organic carbon, the Mackenzie is quite a complex system. The sources of carbon in the river can be microbial biomass, plant debris, mineral-associated organic carbon, and rock-derived organic carbon, which they present different ages. For example, in terms of dissolved organic carbon, it's been mostly modern over time, although in, I think, 2018, it was reported some relatively old signals of organic carbon. In terms of particular organic carbon, or POC, is mostly biospheric carbon is H, is quite old, and this reflects inputs of H permafrost soil organic carbon in the river. So I think this is from Melissa Sharp. I think it was is the most recent kind of compilation of the properties of POC in this catchment, and it shows well that in this system there is a perhaps strong correlation between the age of the organic carbon, as shown by 14C, with the nitrogen to organic carbon ratio. So this ratio, as it increases, is more degraded, and also that shows that the organic matter that is in the river is not only more degraded, but also older in age, which means that the source can be also older. So if we look at the thermal stability of POC, that this is pretty much new data. We've been looking at suspended bed sediments from 2009 to 2019 from 5 locations: the Peel, the Arctic Red, the Mackenzie, the Liard, and the Delta region. And the first thing we did was to generate these thermograms, which I mentioned before. Basically is temperature, CO2 over temperature, right? And here we have all the thermograms for bed load sediments at the top and suspended sediments at the bottom. So as you can see, there are some differences and at the same time similarities across all these thermograms. For example, there's an increase, or the highest concentration of CO2 is observed at nearly 400 degrees, but some samples show like a higher or lower component of organic carbon at higher temperatures. This saying, for example, higher than 500 degrees. And this, the reason for this, there are several reasons for this. It can be like, well, a different source of organic matter, or the different or more degraded organic carbon, basically. And then using the thermograms, we can calculate probability distributions of activation energy. What we have here in the x-axis is the activation energy as a function of probability. And with this, we can calculate also 2 metrics. First is the mean, and the second one is the standard deviation. The mean of activation energy, just to put it simple, as higher values means more stable carbon. And the standard deviation, higher values indicate more chemically diverse carbon. This allows also to compare samples, because they are all they are all standardized. Now, if we look at the differences in activation energy between rivers, we can see some rivers we can see some trends that match what we see in the landscape. For example, activation energies are lower in the Liard, then it goes higher in the Peel, Delta, Mackenzie, and the Arctic Red. And this is suspended sediments, but also Bedloes shows a different sequence with the Delta showing the lower values, followed by the Mackenzie, the Peel, the Arctic Red, and the Liard. Now, if we look at the standard deviation of those activation energies, we can see that in all rivers, the mean activation energy is always higher in the bed loads. However, the standard deviation tends to be lower in the bed loads. So this means that the bed loads show more degraded organic carbon, but at the same time, this is less chemically diverse. And there are two processes happening here that I'm going to discuss in a bit. But they are related to perhaps different sources and different composition of the sediments. But we can also see that the differences between bed loads and suspended sediments are are higher for some rivers, but they are not that different from others. And that shows that, for example, there's stronger hydrodynamic sorting in the Peel and the Liard, just as demonstrated by this activation energy. Now we can look at this because in this river, we are sampling across the depth profile. We can look at the variation in activation energy as a function of depth. Here is in the y-axis the mean activation energy, and depth in the x-axis. In the x-axis the mean activation energy, and in the y-axis the depth in meters. So here the general trend is that as we go deeper, the activation energy decreases. And these different lines are because these different lines shows different sampling campaigns. We've been sampling almost every 2 years for the last decade. And we could eventually show or try to show some temporal trends, but for some rivers, we see high variation in the activation energy, which is kind of expected given our sampling strategy. But if we look at this trend, between activation energy with depth, we can try to understand we can explain that differences using the aluminum to silicon ratio, which is a proxy for grain size. So we have coarse sediments with lower values and fine sediments with higher values. So what we can observe here is that at higher activation energies are present in the suspended material that is finer, or those small particles. When the material is more coarse or is bigger, the activation energy tends to be lower, and this means less stable, and here more stable. But we don't see that trend in the bed loads. We see no correlation basically between activation energy and particle size, which indicates that the material that's present in the bed load is different than that of that we can observe in the water column. And the other thing that we can do is, well, is to look at the correlation between the mean activation energy and the standard deviation of the activation energy. Once again, as the activation energy increases, it means that is more organic matter is more stable, more recalcitrant, and as the standard deviation increases, is more diverse. Organic carbon is more chemically diverse. And we can see here that, once again, the bed loads don't show that, that correlation, but we can observe that correlation in the suspended sediments. So what's happening here is basically two mechanisms. One is organic matter preservation by the presence of minerals in the water column, but selective preservation of organic matter in the bed loads. We can also look at the correlation between the activation energy, or this metric for stability, and the nitrogen to carbon ratio, which is a measure of degradation of organic matter, which higher values showing more degraded organic matter. And that we see once again is that as the activation energy increases, organic matter tends to be more degraded. And we don't see that in the bed loads, once again, because the sources or the material that is in the bed load might be a mixture of plant debris, not necessarily mineral-associated organic matter. And we can see some differences, some correlations with a radiocarbon age that shows that the activation energy increases as radiocarbon age decreases, which indicates that the material that is present in the catchment is becoming more degraded, but is also from an old source, which in this case, or which some authors have suggested, degraded or aged permafrost soils. But definitely we need more isotopic information to better constrain that. We can also use the activation energy to quantify the different proportions of carbon and that are present in these sediments. For example, in these circular plots show the proportion of carbon that is in that presents an activation energy of less than 150, which is more like biospheric carbon, and the proportion that has a activation energy between 150 and 175, which is more like mineral-associated stabilized organic carbon, and that organic carbon that is very stable, probably rock organic carbon that is has an activation energy that is quite higher than 175. And we can see differences across the sides of the rivers, which shows which are related with pretty much the land use and the land use of the catchment. But in general, we can quantify them if we combine them all together. And the Mackenzie, the river sediments in the Mackenzie, they contain 78% of biopheric carbon, 26% of mineral associated or this kind of mixture, more stabilized organic carbon. and 5% of rock-derived organic carbon. Once again, these numbers are estimates, and we need isotopic information to better constrain these, especially the rock-derived and biospheric, which are related, which we might need, of course, radiocarbon information to better assess these fluxes or this composition. So just to summarize, ramp temperature oxidation of river sediments provide insights into organic carbon cycling, in particular in terms of stability and reactivity and preservation. Isotopic analysis at different activation energy intervals are needed to better constrain the sources and age of organic carbon, and ongoing and future research are focused on a wider spatial and temporal assessments. We are looking at other tributaries and recent campaigns in last year in 2023. And I just want to leave you with a picture of our one of the last sample campaign and a QR code if you want to check more information about our project Roofscape and the work we're doing looking at the sources of greenhouse gases across these warming landscapes. Thank you very much.